Okay, so you have removed the water molecule. Now, if you want to know how the protein, how the this NADPH are interacting with protein, how do you know? What are the residue? So what you do? Just you can rename. So you just select a chain. Okay, go to here, or you can select the chain from here also. Just click, go to right window, bottom down, just convert it into chain, and just select it. Okay, so everything is selected here. Now you just rename it chain A. Okay, just rename it. So you just select the NADPH also of this one and just rename it. So A is with glycerol also. There is no need of glycerol, but I just keep here A chain. And these are the four different molecules. One is the glycerol that is present in the A chain of the protein, and this is NADPH. So there are three glycerol and one. Sorry, there are one acetate here, two acetate is present in one chain, and two chloride ion is present in A chain, and three glycerol molecule is present in A chain. You just rename it, go to selection, rename it chain A. Okay, just rename it chain A. Okay, now if you select here, the whole chain is selected. Okay, you can hide it if you want the chain A, just hide everything, everything is hide. Okay, except this acetate is not selected, that's why it is not hiding. I just remove it, there is no need of this one. Okay, in this way you can show the chain if you want, show cartoon, you can show the cartoon. Okay, so because NADPH exists as a stick, so you have to look at do show stick. Okay, in this way you can convert your structure from one part to another part. Okay, like I just shown the structure as a cartoon or as a stick. Okay, so just remove it if you want, just hide the stick. And because the NADPH is not selected, you have to select the NADPH again if you want. Just select it here and show a stick from here. Okay, so here NADPH selected and the acetate ion is also removed. You just selected acetate ion and just show a stick. Okay, now you can see, and one more acetate ion is here. Okay. So one I have removed it, that's why it is not showing here. Okay, so what to do now? I just start working on this B chain. Okay, remove the A chain if you want. I just removing the A chain, just select it and delete it. So I remove the A chain because this is dimer, so there is no what to worry. Okay, now see, this is one monomer of the human zeta-cysteine. Okay, so I just remove this. There is no need of this chloride. I am just removing it. Just select it, just enter it and just remove it. So this is chain B. Okay, now you want to do, just shift your keyboard and just drag the, if you drag this one from the mouse, everything is selected. Okay, now just rename it chain B. Rename it to chain B. Okay, so this is your human zeta-cysteine. Now if you want to look at the interaction, how this NADPH are interacting with the protein, how do you do it? Okay, so there is some option. If you go to this A action, there is some mode called preset. Okay, or there is some option called preset, and then it's called technical. If you go to this technical mode, okay, if you go to this technical mode, you can look at each interaction, how this NADPH are interacting with the protein. So I just select this NADPH and orient it yourself. Just orient it. Okay. Now look at the interaction, how they are interacting. Just select each residue, how they are interacting. Okay. I'm just selecting each residue. See this nicotinamide ring. This is the nicotinamide ring of the NADPH. These are interacting with this residue. These are interacting with this residue. Just select it. Just okay. So you have to change the mode from here. You have to change this to, if you do the edit viewing mode, you can change the mode from here. Just change the mode to residue. Now you just select it. I have selected one residue here because these are interacting with this residue with the help of hydrogen. This yellow dotted are the hydrogen bond. Okay. Now you just selected this residue because these are also interacting with this nitrogen of the. So blue one is the nitrogen, red one is the oxygen. Okay. So look at the NADPH structure. This one is the nicotinamide ring. Okay. This is the ribose sugar. This is the diphosphate. Okay. This is again the ribose sugar. And then this is the adenine ring. Adenine ring of the NADPH. Okay. So where the electron will transfer? This nicotinamide ring. It is involved in the transfer of electron. Okay. So just select the residue one by one, which one is interacting or not. Okay, you just select it here and just orient it toward yourself. If you orient it, then everything is clear, very much visible. See, this is selected, this nicotinamide ring making a bond with this residue, just select it. Okay, so this residue are selected. Now you just rotate your molecule through the mouse. Now see, this ribose sugar interacting with this tyrosine. This is the tyrosine. Just select it. Okay. Now if you want to look at other residue, what they are interacting, so go and move it and select this residue, this arginine. This is arginine. Okay. Now see this in the back side, this is interacting, this phosphate is interacting with this residue. Phosphate is interacting with which residue? This residue. So just select it. When you click it, it got selected. Now coming to this part, this one is also interacting. Just select it. Okay. Now which one is interacting? Look at this one. This one is also interacting, and other one is this one. Also proline is interacting, and which one is remaining? Everything is selected except this one, this residue. Okay. Now you can find the interaction in this way. Now what you do when you select everything, okay, so you can rename it NADPH binding site. You can rename it. What is this? This is NADPH binding site. Okay. Now you just show as stick, because these are the line representation. So I want to show as a stick representation. Just click it here, and you can hide the line from here. You can hide the all protein. You can hide the ribbon or line. I can hide the ribbon from here, and I can hide the line also. Okay. So you can see here, these are the interaction that are the present in the protein itself. But I am not interested in this interaction. I am interested in this interaction. Okay. So now you can see how the amino acids are interacting. Only this much of an amino acid that are interacting with the protein residue. How do you know which residue are interacting? Okay. So what do you do? Select each residue. So you can select each residue, and what you do, just right click, and you label your residue. Label your residues. Okay, go to the setting. You can increase the size of the label from 24 or 18 if you want. Go to again setting, change the font, change it to sans bold. So in here you can look at now which residue are interacting with the protein. So NADPH. So you can change to editing mode here and just press your caps control button from keyboard and just drag it. So in this way you can look at how the NADPH are interacting with the protein. So these are the amino acids that are interacting with NADPH. In case of Homo sapiens, zeta cysteine. Got it or not?
Yeah, got
Hello?
it.
Okay. Now, if you want to look at, if you want to know, if you want to work on this NADPH conservation, okay, let open other structure because we are comparing the NADPH. Okay, we are looking at the conservation of NADPH. Let open some other structure like there is some structure called E. coli. There, that one is also crystallized with NADPH. So I'm just opening the other structure. So what you do now, just select this whole one, or you can save it if you want. So just you hide this polar contact. If you change the polar contact from here, okay. If you want the image in this format, you have to hide this polar contact, okay. Or you have to like I will show this how do you make a figure in this way. Likewise, if this mark right in your copy how they are interacting. This one is interacting with this residue. This one is interacting with this part. This red one is interacting with this blue one. So there are 1 interaction, 2 interaction, 3 interaction and 4 interaction. So just you hide it here. First you make a screenshot of this one, then you hide it. Okay, 1 2 3, 1 2 3 and 4. Okay, go to wizard. Okay, and show measurement. Now you just click it because we know these are the interaction. Just click it here, just click it here, just click it here. So you will get the hydrogen bond. You have to first check, otherwise you get confused. You have to go to this one, which one was interacting, then you have to do it like one. Okay. Now go to the editing mode if you done. Because I missed all this other interaction, you have to do like this way. You have to rotate it and find each interaction. But our main focus is I already know these are interacting. So our main focus is to look at the conservation of the amino acid with respect to NADPH. Okay. You can go if you want, go to the editing mode and then you just click it here and press the control button and move the distance. Because the hydrogen bond fall between 2.8 to 3.2 are used on resolution. Okay. So these are the distance. Mainly hydrogen bond, if you look at the hydrogen bond, it fall between 2.8 to 3.2 and before that, above that, all are the hydrophobic interaction. Okay. So now, if you want, I can do for everyone. Okay, go to this one. Now I have already done this way. Now look at this interaction. Okay, just hide it, polar contact, and then go to wizard, again measurement, and then you just click it here. Okay, and then if you look at other one, what are the other interaction? Which one? This one. Okay, go to again polar contact, go to wizard. These are the interaction. Okay. Now which one is other remaining? These two are the remaining. Okay. These two, go to hide it and then sorry, this here to here and this here to here. Okay. Go to wizard, measurement, and then you just click it here. So these are the interaction. and then you just show it again, which one other interaction. So these two are interacting, these three, these four are interacting. 1, 2, 3 and 4. Okay, just hide it polar contact. 1, 2, 3, 4. This is 1, second, third and fourth. Okay, so these are the interaction. Then go to this part. Which one is interacting here? These two residues are interacting. Okay, these two, or you can zoom it if you want. You can zoom it here and rotate it according to yourself. So these are the interaction. Add it. So it, okay, just add it. Go to wizard, measurement. This one is interacting with this one. This one is interacting with this one. Okay, confirm it. So I have completed all the interaction. You understood this way or not?
Sir, the thing that you are performing, can we get a video or something
Yeah,
so that we can
you get
practice
the,
of our own?
you get the session file.
Okay, sir. It would be
Or,
great.
or you can, anyone can record the whole session. I don't know how to record the session. I think whoever shared the meeting, he or
I'm
she can
recording
record
my
the.
screen.
You are recording the whole screen, na?
Yeah.
Okay. So I have shown every interaction. Which one, anyone is missing? You can look at this one. You can find anyone is missing or not. Everything is intact. Each residue is intact. I have done for each residue. Okay. Now what you do? Just hide it from polar contact here. Now you get this nice interaction diagram. Okay. Now if you want, you just select whole. If you want, so these are the selection. Okay. So these are the selection. Go to the editing mode. Just done it. Go to editing mode. Press your caps lock and just drag it each. What are the distance of hydrogen bond? What are the distance of hydrogen bond? So what I am showing now, it's a very simple way to get the interaction. Otherwise, people are doing other command way. In that command way, you get some, you miss some interaction. Definitely you miss some interaction. But in this way, you get every interaction from the technical way mode. Go to this, click this one. Okay, this way, and then this is 2.9. This is valid. Okay, this one is not interacting. We will remove it. And then okay, this already done. 3, 2.7. Okay, this is 2.9, 2.9. So these are the conventional hydrogen bond. Okay, these are the conventional hydrogen. There is something called hydrophobic interaction that also play interaction, but we mainly focus on here hydrogen bond. Okay, maybe like this one is interacting, making hydrophobic interaction maybe. So this one is not. If I just click it and remove it. Whenever you click it, you have to change it to viewing mode. Otherwise, you can't delete it. Just click it and remove it. Okay. Now you can save any session if you want. Like if you go to your supervisor, and if you want in this orientation, okay. So go to the wizard. Go to wizard, or you can go to scene. There is option called append. You just append it. Now if you want to show this way, like if you want to show in this way, okay, or you can go to your full screen, which if you want to orient this way, in this way every interaction is shown. Or this way I think every residue and interaction is solved. Okay, go to the wizard again, setting wizard or scene, you guys append it second. Okay, now if you want to orient this way, likewise if you want to look at this way, no, go to scene again, append it. Now look at here, you had three different scene. Scene 1, scene 2, scene 3. You can alternate between these three scenes. If you click here, okay, this comes this way. If you click this way, okay, you can, if you click this way, it will orient your molecule according to yourself. You can create lots of scene. Scene 1, scene 2, scene 3, scene 4, scene 4. Okay, now what you do, just select this residue and all this one. Go to, I have selected this residue, I have selected this and then this, okay, this residue, this residue, this one and this one, because these are the active site and ADPH binding site. Okay, now you just rename it. You just, when you click it, you just rename it as what? Just rename it as NADPH binding site. This is NADPH binding site. Okay, this is NADPH binding site. When you click it, enter it, it written here NADPH binding site. Okay, this is NADPH binding site. This is completely selected. Now, if you want to show as a surface view, what you do? If you want to look at the pocket, how the pocket NADPH is present, go to show surface. So see this way, you can find the pocket of the protein. So NADPH, this is the pocket for NADPH, this is the pocket for NADPH, and the residue that are interacting in this way they are interacting. Go to show cartoon. Show cartoon. Okay. Now you can show your pocket here also if you want. So see the protein in a different orientation and selected residue in a different orientation. This one is very much complicated actually, but if you want to show the only protein ligand interaction, then you just hide the cartoon from here. Hide the cartoon. So you can see, you can show this way. Okay. Or you hide the surface from this one also if you want. Hide surface. Hide surface. Okay. Now if you want to export this image, okay, if you want to export this image, how do you do it? If you want to render this image, protein ligand interaction in 3D, there is something called 2D interaction. This is 3D. In 2D interaction profile, you can get 2D interaction profile in Discovery Studio. There is some web-based software called Protein Plus. Believe I will show you. Or even though from one web-based server, if you get the interaction, they will provide you the PyMol session file. You can directly open this type of structure from that PyMol, from that web server. Okay. I am doing here manually. I will show you how to get this image automatically in your PyMol session. Okay. Now what you want to do, if you want to save this image, you just select it according to yourself. This I just select like this way. Okay, because this look good to me. Okay, go to the action. Or if you go select this one and just tell the software to orient yourself according to yourself. So this way I think this is good. Everything so just a little bit shift, I will shift. Okay, now you go to the action, go to draw and just render your protein if you want. Just render it to 300 dpi. Okay, now you change it to 224 and okay, this is fine. Now you just draw fast. If you render it, you get this image. You have to save this image If you want, you can save this image to any file and then you could keep this image in your paper. Okay. So you will get this type of image in your in paper. Okay. Now I want to tell you how the other protein are interacting. Okay. How the in other protein how the NADPase is interacting and what are the conserved residue. That is my focus actually for my review paper. Now what I am going to do, I am just opening some other structure. Okay. So that you can understand about the conservation aspect of NADPase binding site. Like like I can tell you one thing. See this leucine 271. Okay. If leucine 271 is present in other cases and also it interacting with adenine ring, it means that the leucine 271 is conserved in all the cases. If it is present in all the cases. So like I will open some other protein structure. Let's say I will open which protein structure you want to open. Let's say I will open this is 1YV5. Let's say I will open 1QR. 1QR is for E. coli. Okay. I just open 1QR. Now what you do? So this one is already I have done it. Already I have done. Okay. So now once I will get this type of image from this one, I will superimpose these two. Okay. So this one is
Can
this
you
one.
do
You
it
can
with
hide.
this mania QR?
You want to do with this one QR.
Yeah, because it doesn't have this structure doesn't contain any DPH.
And DPH. Okay. So you can say this one is because but you can't do this. You have to dock the NADPH first, and then you have to compare. How do you compare? For that, you have to open the AlphaFold docked LDQR structure. If you want, I have AlphaFold docked NADPH structure.
Okay,
If
you
you want, I
can
can open it.
yeah. I want to see that too.
Okay, so I will just minimize this one. So I just minimize one QR. I will be opening the AlphaFold structure. So what? Wait a minute. I will show you how you use AlphaFold because this time AlphaFold will get the novel prize, है ना? So I will show you how you get the. I will stop presenting. है ना? So I will share the Mozilla. Okay. Now you can see. I tell you. You can see this window. This Google.
Yeah, it's visible.
Yes. Yes. So now open AlphaFold 3. What you do? AlphaFold 3. Okay. EMBL AlphaFold 3. Okay. When you open this one, what happened? Not opening. Yeah. So this one, EMBL AlphaFold protein structure. You just open this one. So this one is developed by Google DeepMind. Okay. For that, they get the Nobel Prize, the David Becker, no, and some other people, two people are, they get the Nobel Prize. Okay. So go to here. There is some link to this one. Google DeepMind. I think this is the Twitter link actually. I think they have written EMBA. Yeah, this is the main web page. Okay.
Okay.
So here what you can do.
Hello? Hello? Actually, I lost the connection.
Who is lost? Only two people are joined.
Hello?
Four, only two have joined. Who have lot of people are lost. Yeah, tell what happened.
No, no, I lost the connection for 1 2 minutes only.
Okay, okay. Somebody have leave the chat, leave the meeting like that, na? Only Shraddha and Susmita called in. What about other people? Where they?
I'm recording the screen. I think they will watch the screen. You can continue.
Okay, so you can see this slide, this window.
Yeah.
So what you do now, you can paste your sequence here. Which sequence you want to fold it. Okay, like this, and look at the how this AlphaFold server work actually. Okay, so when you when you get the result from this AlphaFold, okay, when you get the result from this AlphaFold, okay, and when you superimpose your AlphaFold structure with the solved PDV structure, both are very much superimposed to each other. It means that this AlphaFold is doing very much great job actually. There is no need to crystallize the protein. Okay, now I will show you how to run this job. Go to this one and just input it here. You just input your sequence. So I am inputted the L-Jonathan sequence here. So the PDV ID is CBJ31296. Okay. So this one is the PDV ID. Now just you have to copy paste your sequence here. Go to the download faster sequence. You just copy paste the sequence. Okay. Now you just paste your sequence here. Just you have to click it here. Okay, it's not taken by. This one is not required. Actually, this is not taken.
Actually, your greater than sign is there.
This is not required. No, this is required actually. Now it has taken. Okay. So the sequence is taken. Now what you do? बार-बार message करिए जा रहा है. Okay. Now what you do? What you want to dock to this protein structure. Okay. So what the AlphaFold will do? It will fold this protein. Okay, and then which molecule you want to dock it? Add entity. You want to add ligand, you can just add it ligand because this protein is work with NADPH. So we are looking at because this one is without NADPH. So we are trying to dock this protein with NADPH. Go to action here, go to this one, and then find NADP or NADPH. This is NADPH because this is NAD oxidized form, so not work. This is NADP. This is NADP. This is also oxidized form. This is reduced form NADPH. Okay, you can look at the interaction from both the three. You get the same interaction. No problem. I am just for now I am using NADPH. Okay. Now you select this one. What you do? Continue and preview your job, or you can continue your job. So when you run it, you will get the result where this protein dock your NADPH molecule inside the protein. Okay. Now just you have to continue and preview job. Confirm and submit the job. So this job is already launched. it is running here. Okay, so this will take I think 5 to 10 minutes. So I will show you when you run this result.